He is a PA of our program. He is a plant physiologist, both MSc and PhD from IRI. And after completing his PhD degree, he joined the same institute and is continuing his bond with the alma mater for over 30 years, continuously. Yes, Judge. It is one of the incredible records. He is no well-known stress physiologist. Here, wonderful publication in Nature, PNAS. You name the journal, any journal, his publication is there. He is one of the top cited researchers in the country in the agriculture field. So we are glad to have Dr. Krishnadan for lecture on a kind of a person result. You know, basic and applications in crop improvement. Sir, floor is yours. Thank you.
Sir, here the coverage is sir. Sir, ये लगा लो. Yes. ये पॉकेट में रख लो. आज ठीक है. इसमें जाएगा लेकिन इसमें नहीं जाएगा. आप इनको बोल दो कि यहीं पे रहेंगे. वो हिल रहे थे ना तो मैंने इसलिए
So already we have written about genomics this time. Otherwise, also I think better to several lectures on genomics. It is one of the hottest topic in the recent days, especially after 2021 the Nobel Prize for chemistry has now everyone is interested in this technology. So I will briefly telling you about why this technology is important for Indian agriculture. So, as we know that by 2050 we need to feed about 9.7 billion population. India alone will have about 1.67 billion population in the. So we need to provide food, feed and shelter to the large population for their requirements for various demand. So at the same time, as you know that the climate change is one of the major concerns globally. So we need to reduce the impact of agriculture on climate change. So that's why we have to reduce the emissions. At the same time, we have to produce more. So this means we need about large amounts of rapid technologies to produce sustainably so that we can meet the demand. So this is just an estimate that how much we need to produce by 2047 when we celebrate our 100 years of independence. So in the footprint, if you see, we have almost from 330 today to 550 billion. About almost about 1.6 we need in production. Similarly, in particles and crop, which is even more, we have to produce almost 2-fold increase in the production. And if you see the other like milk and other available between 6 and 7, which means that we need to increase the food and also we need to increase the feed. And in addition to that, biofuels is one of the important areas. Government is trying to enhance the biofuels So currently, only about 5% biofuel is so by 2031 we have to reach 20% use of biofuel fuel that we use for so which means we need a lot of biofuel. So the main portion of biofuel is definitely the sugar molecules, rice grains,
थोड़ा वाइड करते हैं तब पता लगेगा कि वाइट तो कर वाइट वाइड वाइट इतना नहीं है इधर
genetic gain in the crop improvement when it comes to crop improvement. At the same time, the crop genetically or the resources. So today we are talking about genomics, the improvement of genetic. So the genetic gain currently about 1% right being movement. So it is the more major crop that contributes about 66% of the calorific demand of the global population. So this was the genetic average genetic gain is only about 1%. But this needs to be increased to 2% by 20%. So to achieve the genetic gain, there are different components needs to be addressed within the selection engine. So that is, if you have a population and the potential select that individual, how good phenotype versus the population perform. Which means you have to have in the population a phenotype, good phenotype that can perform better in the face of infection resistance or actually. So that kind of variation you can create by using the the selection accuracy can be improved by modern phenotyping. The genetic variation again we can create by using phenotyping, which is method. The other important part is time factor. So conventionally, we use the conventional breeding approach. It takes about 12 to 15 years to develop a strong variety. And if you use molecular breeding, it takes again about 5 to 6 years. And one of the limitations of the molecular breeding is when you insert the gene of the there are other links between the getting to the allied germ. So there is like alleles which are not available. Those alleles will have to keep accumulating and developing into markers. So in case of genotyping, this problem is not there. We need not to bring other alleles because this allele is genotype that is the performing better. We can set it to into one gene at a time. The genes of interest at a time. So you are not going to alter such a genome combination. So there is no linkage drag or any negative elements going to circulate. At the same time, accuracy improves this which can take about 3 to 4 years is sufficient to improve existing variety. So you reduce the reproduce the portion and you reduce the gestation time while you can use this genotypic variation so that the genetic gain can improve by matching innovative. The same goes innovative is new method.
It depends upon the mutation. So just to show the power of mutation, I have put these two pictures. Noble laureate Norman Borlaug standing in the wheat field with the semi-dwarf wheat that he has created versus the normal wheat which existed before Green Revolution. So this is only a single gene mutation. So can anyone say what is this mutation? So in what gene mutation was there in this one? In the this reduced height one gene is codes for what protein? What gene it is? So Norrington is the donor name. So what is the name of the gene? What it codes for? So so if there is mutation, the plant is dwarf. So a gene should code for some protein or some RNA, that function. No? So what this gene codes for? This they are depending upon the interaction of the protein DNA interaction. That is, they used proteins which can bind to the DNA at specific places. And the first one was the endonuclease, forming endonuclease, which is also called as meganuclease. These are coded by the organism itself. Only if you modify some amino acids in the meganuclease, you can target this nucleus to a location which is other than the native location. But again, it is depending upon the engineering of amino acid. You have to engineer several amino acid residues to target the protein from the native location to your target location. And it is also not possible to target this nucleotide to all the locations that you are interested in the genome. Then an improved version came, the zinc finger DNA binding protein plus combined with the endonuclease. So again, this zinc finger also you have to assemble multiple zinc finger to target this assembly to a specific location. Again, lot of protein engineering works were involved, and it is not easy to target. Then the third approach came, which was transcription.
So the transcription activator effectors are produced by so these are transcription factors produced by bacteria. And this bacteria transcription factors are increased in time. And these transcription factors go on bind in post and it is so scientists are monitoring
the tail protein as a repeat region, which decides where this protein goes and binds into the genome. So you can modify these repeat regions in the tail effector so that you can target this protein to a specific location in the genome. And again, this protein need to be fused with an endonuclease so that wherever this protein goes and binds, the nucleus will make a double-stranded cut in the genome. That is, it induces DNA damage at specific locus. Then during the natural repair process, some mutation arises. So all these three technologies, they were using the principle of DNA protein interaction, and you have to engineer the protein so that these proteins are targeted to a specific target loci in the genome. Since there were a lot of protein engineering were involved, many labs could not use this technology. Then Professor Jennifer Doudna and Emmanuel Carpentier, they came up with a technology wherein an RNA molecule was used to guide the nucleus. That is, the nucleus is taken to a specific genomic location by an. So it is very easy to design RNA molecule, because as you design primers, you can design this RNA molecule which is about 20 nucleotide in length, and which can be used to guide the nucleus, which is the Cas9 protein, to a specific location. So that's why this technology has become, after its discovery in 2012, it has become very popular, and by 2015 already it was declared as breakthrough of the year, and then by 2020 these two discoverers, they received the Nobel Prize. So this was based on the immune system that the bacteria has. This CRISPR-Cas system is based on the immune system that bacteria uses. So in bacteria, whenever a phage infect, the phage sequence, a specific region of the phage genome sequence is copied into the CRISPR locus into the bacteria. This is a small sequence, about 20 nucleotide is copied into the CRISPR loci in the post genome. And then the CRISPR loci, that is, once it is copied, it becomes part of the bacterial genome. So whenever next time the phage infect, this region, that is CRISPR region, get transcribed, it produces a CRISPR RNA, and it also produces a trans CRISPR RNA, and then it produces the several nucleases are there in the Streptococcus pyogenes, from which this system was developed. So one of the nucleases Cas9 nuclease. So once this trans-CRISPR RNA is produced and the CRISPR RNA is produced, the CRISPR RNA and trans-CRISPR RNA, they form a hybrid like this, and some of the nuclease, they cut this into produce interfering RNA. So this consists of the protopacer sequence, which is from the, and this is the scaffold or trans-CRISPR RNA sequence. So now this hybrid can bind to the Cas9 enzyme, and the Cas9 enzyme search the incoming FOD genome wherever this protopacer DNA is complemented to RNA is complemented to the FOD genome. In that place it binds and it makes double-stranded DNA cut. So that thereby incoming FOD is deactivated. So Jennifer Doudna and Emmanuel Charpentier, in their paper, what they have done is they synthesize the scaffold RNA or the trans-CRISPR RNA, and they the protopacer sequence, that is the guide RNA, in a single guide RNA molecule. In bacteria, we have seen there are two different RNA molecules are there. They form hybrid, and then it is processed to produce the guide RNA and scaffold RNA complex. So in this case, if you make a single transcription unit consisting of the guide RNA plus the scaffold RNA, this can be transcribed as single guide RNA. And then if you use along with this one, if you use a Cas9 enzyme, then this complex can go and target the region of your interest. So this was demonstrated by Jennifer Doudna and Emmanuel Charpentier in bacteria, and then later Feizan, he demonstrated this in the genome editing in human cells, and later this technology has become very popular. So from 2013 onwards, you have seen a lot of publication in the plants. So in the genome editing, you need two reagents you have to use. One is the guide RNA, which is complemented to the target loci. So just like you are designing a primer to amplify a specific region in the genome, the guide RNA is designed to target, take this molecular scissor or the Cas9 or other version of the Cas12 to a specific location in the genome. So you need two reagents, and then these two reagents can be delivered into the cell as DNA, or it can be delivered as RNA plus protein complex. That is, the single guide RNA molecule can be provided as RNA, and then the Cas9 nuclease can be provided as protein. That is, if you mix this single guide RNA plus Cas9 protein, and if you deliver into the cell, it can go and create mutation in any of the target region that you have, for which you have designed the single guide RNA. So if you want to deliver the reagent as the RNP particle, that is RNA plus protein particle into the nucleus, it is very simple. You do an in vitro transcription to produce single guide RNA, and then you produce protein by using any of the overexpression system in the bacteria, isolate the protein and mix it with the RNA that you have synthesized, and then you can deliver into the cell. So this one is easy for the human or for the protoplast. In case of plants, delivering the RNP particle into the cell is very difficult because plants have the cell wall. So we have to mostly use it as a DNA, which is the most common method of delivery into the plant cell. So if you want to deliver these two reagents in the plant cell, the guide RNA is cloned under the RNA polymerase driven promoter. So RNA polymerase produces the guide RNA, and the Cas9 protein, which is from the Streptococcus pyogenes, which is a prokaryote. So you have to optimize the codon for the eukaryote. For each plant, some people optimize, some people use even the Cas9 optimized for human genome. That also works well for the plants. And then you have to add nuclear localization signal. That is, this protein has to be taken into the nucleus in case of eukaryote. So for that you need a nuclear localization signal. So whenever you buy a vector from AdGene or anybody, this basic structure will be there. Only you have to clone the guide RNA just at the transcription start site of the RNA polymerase 3. So only if you do the simple cloning here, then rest your entire cassette is ready, and this can be delivered into the plant by using either agrobacterium mediated transformation or by using protoplast transfection. So once these reagents are delivered into the cell and the guide RNA is produced and then Cas9 enzyme is produced, they form a complex, and this complex searches the complementary region which is complementary to the guide RNA. And only one condition is that it should have a PAM sequence. For the Cas9 nucleus, the protophasor adjacent motif sequence would be NGG. Any nucleotide can be there followed by 2 Gs. So the Cas9 binds to the PAM site, and then the guide RNA makes a complementary base pairing with the target sequence. Once this complementary base pairing is established, the Cas9 cuts the DNA to make a double-stranded break in the host DNA. So you get a double-stranded break in the genome. So any DNA damage is there in the cell. So till cell has top priority to repair the DNA damage. There are several repair mechanisms that operate in the cells. One of the native repair mechanism is non-homologous enjoining. That is, these kind of breaks are there. The cell will do something to join together these two broken in. So during this repair process, there may be one nucleotide addition, or there may be few nucleotide deletion. So these kind of repairs, which is mediated by the non-homologous enjoining, which is unguided repairs. That is, we target the Cas9 and guide to create only the double-stranded break in the target loci, wherever the mutation has to be generated, and then the cell takes its own course of action to repair this double-stranded cleavage site. So during this repair process, chances of mutation is there. That is, it can be a small insertion or it can be small deletion. So you get mutations. So these kind of mutations are called as the side-directed nuclease 1 mutation. So where after the DNA cleavage is happened, there is no guidance is given to the cell how this site should be repaired. So this is the SDN1 mutation. So in case of other type of mutation, which is called as SDN2 mutation, wherein you guide the cell how this cleavage site should be repaired. So for this, you provide a guide template. So you provide a DNA template which is complementary or homologous to the target site, except for one or two changes. That is, you want to make one nucleotide or two nucleotide or specific changes. So in the guide DNA template, you add those changes, and the rest of the region is same as that of the native place. Then by the homology dependent repair mechanism, this template helps in the repair of this particular region. That is, this region is replaced with the broken region. So now you get a repaired DNA template with only the one nucleotide change that we introduced. So in this, we have guided the cell how the repair should be taken. These kind of mutations are called as SDN2 mutation. So this SDN1 and SDN2 type of mutations, they are exempted from the rules 1989, that is GMO regulation rules in India. So if you develop any mutants by using this technology, and if you show that the reagents that we originally used are absent from the mutant that you finally generated, then these two are exempted. You can use them as a normal mutant. The third type of mutation is the SDN3 mutation. So in case of SDN3 mutation, what we do is we have the homology arms, that is sequences which are flanking the cut site are included in the repair template, and in the middle of the sequence we add any foreign gene. So if you want to introduce any foreign gene, either from the same species or from the different species, we can include the foreign gene in the middle. So get integrated into the cleavage site, and then you have the foreign gene in the new foreign gene is integrated into the genome at the specific target loci. So this one is mainly useful to target the foreign gene into a specific locus into the genome. So normally when we use a transgenic approach, the foreign gene goes and integrate into the genome and random location. So depending upon the location where it gets integrated, the expression will be different. That is, if it is in the heterochromatin region, it may not be expressing, and if it is in the euchromatin region, depending upon the region where it gets integrated, the expression will be different. That's why if we generate 10 different transgenic events, the expression of everything will be different. Used 30 base promoter and you clone the gene of your interest in front of that, but still you will see a lot of variation in the expression. So those kind of differences will not be there if you use the site-directed nuclease 3 approach. And also when you use the genetic transformation by using the conventional transgenic approach, the tDNA can go and integrate into the any location on the genome. So which again can create mutation. That is, if it is integrated on the gene of any gene of interest, or any gene that has any role in the plant. Of course, all the genes, they will have some or other role in the plant. So they will go and integrate into the gene. So the gene get disrupted. So you get a tDNA mutant. So if these kind of mutations are there, then those events will not be useful. That is because already there is a mutation in some or other. So those possibilities can be avoided by the SDN3 approach. And later some improvements came in. So here these approaches, they were mainly depends upon the creation of a double strand cleavage in the target loci, and then repair, either unguided repair or guided repair was used to create this kind of mutation. So a third, the next kind of mutations creating approach is known as the base editing approach. In case of base editing, what we do is we don't create a double stranded break in the only we in the target, and the Cas9 enzyme is fused either cited in DMA, depending deaminase in deaminase Cas9 enzyme. So guide RNA is same as that we used in the earlier approach. So in this case, your target region should be about 10 to 12 base pair upstream to the PAM site. So within the target region, the deaminase, this region will be inside RNA complex. So you target a region which is about 10 to 12 nucleotide upstream. So by this method, you can C to T substitution or A to G substitution mutation can be created. Only you just want to change one nucleotide, so that is possible by base editing. Another improved version of base editing is again the prime editing. The base editing frequency is very less, and only you can to change either the C or A, as I told earlier. So in this prime editing approach, you can change any nucleotide. You can change little bit larger modification. You can introduce a short deletion, introduce a short insertion. So everything is possible. So in this, again, in the those two reagents that we discussed, the guide RNA, again it is modified. To the single guide RNA you provide at the end of the 3' end of the single guide RNA, you have the editing template. So you include an RNA sequence complementary to the target with necessary changes that you want to incorporate. So in this one is called as, and the guide RNA in addition to the complementary region, this has additional template which will guide where the what kind of mutation that has to be introduced in this site. So this one is called as the prime editing guide RNA. This is the one modification that we make here. And another modification is that along with the NIC Cas9, you use a reverse transcriptase enzyme. In the base editing, we used deaminase, DNA deaminase. Here we are using reverse transcriptase. Case when these reagents goes into the cell, there is a in the and at the cut site, the editing template form a base pairing to make a double strand this one DNA RNA hybrid. So now this RNA strand which we have introduced will be copied by the reverse transcriptase to synthesize new DNA strand at the cut location. So because of this new sequence that is synthesized, again this RNA strand will be replaced by DNA strand. So in the cut region you will have either new sequence whatever you have introduced, or if you have introduced any deletion, that deletion will be there. So with this way you can guide the double strand this DNA damage site or the target loci with RNA. So here we don't use DNA for guiding. In SDN2 we use DNA for guiding. Here we use RNA for guiding the repair process. So these mutations are also exempted from the GM rules. So another important point that I want to say is whenever we talk about the genome editing, so what kind of mutations that can be created. So in case of mainly we think that either some small deletion to knock out the gene or 1 or 2 amino acid modification that can be made. So besides this, there are several possibilities of modifying the gene expression and gene activity. So if you take a gene, it will have different elements. So this I have given for a protein coding gene. So it will have a promoter region, and it will have a 5' UTR, it will have a exon, intron, and then again 3' UTR. So if you want to modify the expression of the gene, so you have a gene of your interest, so that you want to keep expressing in all the stages. In one particular stage you don't want that gene to express. So or in one tissue you don't want that gene to express, especially in case of susceptibility gene or genes that code for senescence. You want them that these genes may be having important role in other developmental stages. Only what at a specific stage you don't want that gene to express. So in case of the pathogen infection, if it may be a bacteria or fungi, if they induce any susceptibility gene, otherwise that gene is required for normal function. So in case of the bacterial blight, the sweet genes which codes for the sugar transporter, they are induced by the Xanthomonas oryzae. So this induction happens because they produce a specific plant specific transcription factor. They go and bind to the promoter region of the susceptibility gene. So if you create a mutation in the cis element that is responsible for recognition of the bacterial transcription factor, then this bacterial transcription factor cannot bind to the gene of the susceptibility gene. So now the gene will express in the normal way in all other conditions except when the bacteria is infecting the plant. So now the susceptibility gene is not expressing, so it can gain resistant to the bacteria. Similarly, if you want to express some genes under the stress condition, that is, you know that this gene expression either get reduced, or if you express a specific heat shock protein, it is going to protect the cell. So you can introduce the heat shock element, that is there again the cis elements can be introduced into the promoter region. So those genes which are normally not expressing under the high temperature stress, you can induce them under the high temperature stress. Similarly, you can put any cis element that is responsible for driving the expression, or you can knock out some of the cis element which is causing the expression, so that you can modify the expression of the gene. And the next gene element is the 5' UTR region. So the 5' UTR region in many eukaryotic gene, it codes the 5' URF, that is the upstream ORFs. So these are small open reading frames which codes for few amino acid in length. So actual open reading frame may be starting little further. This codes for the actual protein from this ATG to this TG. But this 5' UTR will also contain some small ORFs. So normally when the ribosome they start translation, they bind to the from the 5' UTR region, it will bind, then it will search for the initiation codon, and the ribosomes, when it comes to the ATG, first ATG codon, it will start the translation process. So because these ORFs are very small, it will have a termination codon here. So after like translating few amino acids, the ribosome will dislodge, and then this process will continue. The futile axis will continue. Instead of going from here to here, this will get translated. So this reduces the amount of protein that is produced from this messenger RNA. So if you delete this URF region, that is only the ATG codon, if you modify this, then the ribosome will start the translation from the actual ATG codon. So this will lead to enhancement in the amount of protein that is transcribed. Other than that, in the exon, you can do different kind of modification. If you think that modifying one amino acid is going to convert into the constitutively phosphorylated form or unphosphorylated form, that you can do. Or you can also alter the motifs that are responsible for protein-protein interaction. So the protein is interacting with one kind of protein, not interacting with other kind of protein. Also, you can modify the regions that are ubiquitinated, so that you can prevent the protein degradation. You can increase the half-life of the protein. So many kind of modification is possible within this exon. So any microRNA binding domain is there within any part of the gene, that you can modify the microRNA target region. That is, you alter a few nucleotides. The coding sequences, the codons are not modified, only just modify one or the third nucleotide of the codon, so that it still codes for the same amino acid, but it will not be complemented to the microRNA. So the microRNA mediated cleavage can be prevented, so that it accumulate more and it can produce more protein. And also in the gene body, there can be several kind of methylation. Either it can be in the promoter region or it can be in the coding sequence. So methylation in the promoter region, normally in plants, this results in a reduction in the expression, and methylation in the coding sequence, it normally promote the expression. So you can do any kind of modification in the, that is the cytosine is methylated, so you can modify this C. The target Cs which are methylated, so you can change the epigenetic mutation can be created in the gene. So all kind of mutation can be created, so you can do any kind of modification that is required for the cell. And the next important aspect is whether the genome edited mutants are GM or non-GM. So if you use ribonucleoprotein to induce mutation, they are non-GM, because there is no foreign gene is going to integrate into the genome. You are using only RNA and the protein. If you use DNA as a reagent to edit the genome, so the DNA will be there and it will go and integrate into the genome. So this is a normal transgenic route where you do a genetic transformation and foreign DNA goes and integrate into a locus into the genome, and then you get the regenerated plant. By selfing, you get a transgenic plant which is having the homozygous to the foreign gene. So this transgenic plant will have the foreign gene integrated stably into its genome, and this is necessary to give the trait of your interest. So if you are developing a this BT transgenics in any crop, so the cryonase gene has to be inserted into the host genome. The foreign gene is required for the expression of the trait. Or if we are using RNA approach also, the foreign gene should be there, and then it is transcribed and it goes and suppress the gene of our interest. So in any case, the foreign gene will be there in the commercial variety, and it can be detected by molecular analysis. If you do molecular analysis, you know whether it is a transgenic plant or non-transgenic plant. In case of genome edited plant, by using the agrobacterium or particle gun approach, when DNA is delivered, again a transgenic plant is produced. And this transgene, once it gets integrated into the genome, it produces the guide RNA and Cas9, and this reagents go and create mutation in the target loci. During the selfing process, you can segregate the transgene separately and the mutation separately. So now this one, normally you will discard the plants that is containing transgene, and you will use only the plants that is containing the mutation. So you get a mutant loci, and the commercial variety will not have any foreign gene, only the mutation that you created is there. And you cannot use any molecular technique to identify whether this mutation was spontaneous mutation or induced mutation or the mutation that is created by the CRISPR-Cas. So this mutation is similar to that of the natural mutation. So that's why these plants are considered safe, and they are exempted from the GM regulation. So there are several countries in which the genome edited plants are considered non-GM, and they are exempted for commercial cultivation. So this is one of the first genome edited plant that was approved for commercial cultivation, and it is produced and marketed. So this is available in the market from 2009. And this is soybean plant that is edited, genome edited, to produce high amount of the oleic acid, not the linoleic and linolenic acid. So this is required for high frying application. And other mutant that is available commercially is the GABA-rich tomato plant, and again this is commercialized in Japan. So this is again available in the market from 2020. And Japan has again it commercialized the fish that are edited with either leptin receptor gene or myostatin gene edited fish. These two are also commercially available. With less speed, less time, they can produce same amount of biomass. So this is also available from the market from 2021. There are several examples of editing in different crops. So some of them I have listed here, you can see later. So besides producing, editing the normal varieties to develop product quickly, you can also convert the wild type into a cultivated plant at shorter duration, which is called as the de novo or accelerated domestication. This is an example where the wild tomato is Solanum pimpinellifolium is edited with different genes. So to produce the almost plant which is similar to that of the cultivated plant. So if we edit 4-5 genes, produce convert a wild type gene into a cultivated plant. So if you edit any domestication genes in the wild type plants of your interest, you can convert the wild type into the cultivated plant. So wild type plants normally they have abiotic stress tolerance and biotic stress tolerance. Normally they have less yield and other non-desirable agronomic characters. So if you edit few genes, you can easily convert those wild type plants into cultivated plants. And also you can create some of the novel products. So normally vitamin D, we think that we need in the vegetarian population, vitamin D deficiency one of the major issue. So by editing on single gene in the tomato plant, the tomato plant that can produce vitamin D has been generated. So we need not go and stand in 3 hours. You can take this tomato, then you will get the this vitamin D. You need not go for non-vegetarian diet. So also the provitamin A deficiency is one of the major problem. To address this issue, the National Agri Biotechnology Institute, they have developed a banana plant which can produce provitamin A. So if you take this, the provitamin A deficiency can be averted. But this plant is because banana is a triploid plant and vegetatively propagated. Here you cannot remove the foreign gene. So that's why they are trying to use the RNP mediated transformation. They have standardized. Soon they may come up with the product with non-GM approach. So in our lab, we started the genome editing work in 2018. So we work on different crops, like mainly we work on mustard and rice. Today I'll be one or two slides I will show about the work that we have done in the rice. So our interest is to increase the abiotic stress tolerance of rice, because rice is one of the cultivated in the largest area among the food crop in India, and it is highly sensitive to various abiotic stresses. So we selected this particular gene, drought and salt tolerance gene, which was identified by EMS mutant screening by a Chinese team of scientists in 2009. So they identified an EMS mutant after screening about 270,000 EMS mutant. They identified one mutant which they named it as DST. By map-based cloning, they found that it codes for a C2H2 zinc finger transcription factor. And if there is mutation in this gene, that is if it is non-functional, you get a plant which are more tolerant to this one osmotic stress, more tolerant to NaCl stress, and also more tolerant to drought stress. So we also attempted whether these kind of mutation whether we can obtain by screening the EMS induced mutant population. We had a EMS induced mutant population of Nagina 22. The project was led by Dr. T. Mahapatra and the former Director General ICAR. When he was there in NIPB around 2007-8 to 2012. So in this process, we try to identify a mutation which can be there in the DST gene, but we identified some mutants which are tolerant to drought, but we could not identify any mutation in the DST gene. So even if you know that mutation in a specific gene will give rise to a specific trait of your interest, it is not easy to by using the EMS induced mutation to get the desirable mutation. So this we started by using genome editing technology. We used two different guide RNAs to target the mutation to different domains in the protein. So we created a different mutation, that is in the guide RNA 1 and guide RNA 2. There were mutations which ranged from a 360 base pair deletion to 2 deletion. So these mutants were evaluated in the field condition and also in the greenhouse condition. So for salt stress, we evaluated this mutant, because in the original DST mutant it was evaluated only in the seedling stage. So we wanted to know whether this mutation can provide tolerance to the reproductive stage, which is very much important in case of rice. So we grew the wild type plant and the mutant plant in the same pot, and then these pots were irrigated with sodium chloride, and then after that the plants were recovered to see how much grain they produce, and we found that this mutants can produce more number of grain as compared to that of the wild type plant, which indicates that these plants are more tolerant. Then we also examined under drought stress condition whether these plants can produce how much grain. So under the normal condition also the mutant produce more grains per plant. Under the drought stress condition, again they produce more grains as compared to that of the wild type plants. So again it shows that they have both drought and salinity tolerance at the reproductive stage. We also carried out this study in the field condition in the transgenic network to see if the drought tolerance can be replicated in the field condition. So in this case again we found that the soil moisture stress in all the fields, it is an average, it was almost similar in all the plots. The relative water content was better in the mutants, that is the mutants maintain better leaf water status. And if you see that grain yield also, the mutants produced higher grain yield as compared to that of the wild type. And again, if you see, depending upon the kind of mutation, the kind of allele, there can be variation. So even in genome editing also, depending upon the gene of your interest, you have to develop multiple alleles so that you can select the best allele from the edited lines. So these mutants also, they produce higher grain yield under the field conditions. So these were once you have the mutation, now you have to take it to the field. So normally for GM crop, you have to take approval from the IBSC for carrying out research. Even for genome editing research, you have to take permission from the Institute Biosafety Committee. And then the RCGM, once the Institute Biosafety Committee approve, then you take RCGM approval for carrying out the research process. If you are developing a product, then the Institute Biosafety Committee gives approval for your research. Then RCGM gives approval for the further field trial, which has to be approved again by GESC. So it will go to these two committees for the field testing, and finally GESC will give permission for the environmental release. Whereas in case of the genome edited plant, the IBSC has been given full power to exempt the mutants that have been generated after informing to the RCGM that we have followed the due procedure. So for this, the guidelines were published by DBT Government of India, and the regulation was enacted by the Ministry of Environment, Forest and Climate Change, which is the ministry that give approval for any environment release of any GM crop. So they also exempted the genome edited plant. If you show that the SDN1 and SDN2 mutants, which are free from the exogenous introduced DNA, that is when we use DNA as a reagent to induce mutation, we have to show that the final product that we develop is free from the introduced exogenous DNA. To demonstrate how the mutant should be analyzed to show that it is free from introduced exogenous DNA, a standard operating protocol was also published. So this, according to this protocol, we have to show that the mutant is sensitive to the selection marker if we have used any. So in this case, we developed our mutant, that is genetic transformation was done, and the transformants were selected by using hygromycin. So in this case, we have to show that wild type plant that we transformed and the final mutant that we obtained, they are sensitive, whereas the initial transgenic plant that we developed, from which the final mutant was derived, it should be resistant. So we have shown that the final mutant that we developed, the sensitivity is same as that of the wild type. Then we have to show that it is not containing any foreign DNA. So for this purpose, we need to design primer spanning the entire vector that we use for genetic transformation. In this, there are two conditions. One is that each amplicon should be about 500 base pair or less, and the overlap between the amplicon, it should be about 50 base pair or more. So for this purpose, we have designed about 49 primers to cover the entire vector region. And then we used these primer pairs and 4 different kind of DNA template to show that the mutant that we have finally developed is not having any foreign gene. So in this, we use wild type DNA. Wild type DNA spiked DNA, that is the original vector that we used to transform is spiked into the wild type DNA. So this is added as one template. Another template is the original T0 transgenic plant you developed, from which the genome edited mutant was segregated. So these these 4 templates were used, and we should see that there should be no amplification in the final the mutant that we have. So this was demonstrated, and finally this mutant got exempted from the rule 7 to 11 of the GM regulation rules, the field trial in the country. So these are the first set of mutant that have been from the rules 7 to 11 of the rules 1989 under the genome editing guidelines. So these are in the second year field trial. So hopefully by end of the beginning of the 20, I will be able to release the result depending upon the. So based on the initial the program that we to establish genome editing capability at ICAR, and we demonstrated that we can develop mutants that are agriculture. We submitted a proposal to the government of India, and the government of India has given rupees 500 crores to the ICAR for genome editing. So this 500 crores are used by almost 53 ICAR institutes. So in crop science division, about 24 ICAR, and institutes of animal husbandry, all working in the genome. This program is on 50 products that are for agriculture. So to summarize, that genome editing is a very powerful tool. So you can develop mutation of for any trait of your interest period of time. So this is required technology for achieving the sustainable developmental goal and achieve and beyond. And it is also very important tool. So if you want to this is the best way. Earlier we have limitation in altering the function and constated function. So this one is very important functional genomic tool which can be on the basic and strategic research, and this technology is under the IP rights of University of California, Berkeley, and the Broad Institute. So if you want to release any command, you need the freedom to operate from. For research license, if you are from the government institution, you need not take any permission. But if you want to release any, you need to have freedom to operate. So the government is in the process of getting this license for agriculture. So the thing is that you should know what gene to be edited, what trait you want to improve. So for everything you have to make critical assessment and decide. So that's why it is the research question that is very important, and it is the question that gives you the Nobel Prize. After that, you can add lab coats, coffee, and air conditioning. That is, if you want to do editing, it is a simple process. But before doing editing, you should know what gene to be edited, which crop to be edited, and what kind of edits to be created. So a lot of basic understanding is required. So we should be very clear about that. So other than that, this technology is all the you need flow, autoclave, and I think that's all. Rest all any simple molecular biology can do the genetic transformation and get genome editing. So with this, I thank the funding agencies, especially the National Agriculture Science Fund (ICAR) and DBT Government of India and ICAR IRI, and the students, those who have done work. I presented work that was carried out by Dr. Santosh Kumar. There are several students and co-PI. They are involved in a lot of work that we are doing, and all the ICAR and IRI authorities who helped in the funding and mentoring. And one more quote I would like to tell you that water is a very precious resource, and it is one of the most important food for agriculture. So almost among the total water use, we use almost about 80% of the water for agriculture purpose only. So we have to use water more judiciously, as the freshwater scarcity is emerging as major problem. So two Nobel Prizes are waiting for you. So one for science, that is if you develop crops that use very less water. So about 98% water that we use in agriculture, even if it is used in the transpiration, it directly goes to the atmosphere. Only that less than 1%, 2% water is used in the biomass production. So if you can reduce that water, and also the better plants, then for science you will get some Nobel Prize. And water is cause for interstate disputes, and maybe in future it will be very critical issue. So you will get one prize for peace also. So with this, I thank you all for your patient listening. If you have any questions, I am free to take. Thank you.
Any questions from anybody?
New species. So, very good question. So, I think for new species, when you make mutation, you have to create so many modifications. Maybe if you use synthetic biology approach, like we use large genomic variation that you need to create. So here we are targeting only few genes. So by using gene editing to create new species, it will take lot of time. You can create, I think, but you have to modify whatever modification you think. Like if you compare E. coli versus the Xanthomonas, and if you want to convert E. coli into Xanthomonas or Xanthomonas into E. coli, you have to need to do lot of modification. So by using genome editing, it will take several years. Maybe by using synthetic biology approach, you can do it faster.
That's a very good question. Create a new mutation.
New species.
But I was just trying to see how many millions of years you want to do the experiment, not like this.
But you can attempt. If you do that, you will get paper in Nature.
Very interesting.
So if there is no question, thank you all. Hope your training is went well. Almost in the penultimate day of training. So thank you very much. Okay, and 2 days more. Thank you.
Okay, so I'm sure the lecturer was sitting and it was very informative, basic to application. So here comes how the technology can play a very important role in solving vital issues. This is already online. You can listen to this lecture again and again, so that you will get more clarity. Because in 1 hour lecture, sometimes you might have missed to understand certain points. How these things can be clarified out. Okay. So with that, I think we'll give a big thank you, sir, for giving the answer. Just to honor the speaker, the protocol. So I think the lectures are going on well. So now with that, the forenoon session we are concluding now. Now you break for lunch, come back to Discovery Center sharp at 2:50. So let us continue the practicals, whatever we stopped yesterday. So let's meet once again at 2:50 in this course.